Okay, let's dive into a fundamental mystery. How does life, life as we know it, behave when you take away its most constant companion, gravity?
And what happens when you expose it to radiation levels far beyond our protective atmosphere?
Today, we're journeying back in time. We're exploring the groundbreaking, sometimes really difficult history of space life sciences experiments,
Mhm.
specifically those from NASA Ames Research Center between 1965 and 1990.
Right. And our guide for this deep dive, it comes straight from the source, excerpts from Life into Space. It's a publication documenting these exact experiments.
Including some really fascinating and actually crucial collaborations, particularly with the Soviet Union right in the middle of the Cold War.
Yeah, quite remarkable stuff.
So our mission today is to unpack this history, not just as a timeline, but to really extract the core insights, understand the sheer ingenuity involved, and see how these foundational steps, well, how they paved the way for everything we know now about supporting life off-world. It's about understanding the why behind the what.
And this isn't just historical curiosity either.
Yeah.
What makes this deeply relevant to you, listening right now, whether you're into space or biology or even just general health,
Yeah.
is that these experiments, they revealed fundamental biological principles.
Mm.
The challenges of spaceflight, maybe surprisingly, offer these profound insights into health issues we face right here on Earth.
Like what specifically?
Things like bone loss, osteoporosis,
Wow.
muscle weakness, like from being stuck in bed for a long time, even cardiovascular changes.
Absolutely. So get ready for some truly surprising findings, maybe some aha moments about how everything from tiny cells and
The
plants
gods and monkeys.
became pioneers, pushing the boundaries of our understanding of life itself. So let's start right at the beginning. What were the absolute biggest challenges the space environment threw at life that they needed to study first?
Well, the sources make it really clear there were two dominant environmental factors, totally unlike anything life evolved with here. First, microgravity, essentially the near absence of gravitational pull. This removes that constant physical loading that Earth's gravity provides.
Right, the constant weight.
Exactly. And second, space radiation, which is both at much higher levels and composed of different types of particles. You've got charged particles, neutrons, ionizing photons, very different from radiation on the ground.
And why couldn't scientists just simulate all this down here? Why go to all the trouble, the expense of actually going to space?
That's a critical point the source really emphasizes. Ground-based labs, they simply cannot reliably replicate either microgravity or the full spectrum of space radiation effects, especially not for long periods.
Okay.
But maybe even more importantly, they can't reproduce the synergistic effects, how microgravity and radiation might interact in complex ways.
Ah, the combination.
Right, ways you just wouldn't see by studying them separately. Space offered the only true laboratory for that kind of investigation.
So space wasn't just a location, it was kind of a unique biological probe. And you mentioned these surprising links to Earth health.
Exactly. The extreme conditions of space, they turned out to be powerful models for problems right here.
Yeah.
The rapid bone decalcification seen in microgravity, for instance,
Yeah.
that gave scientists an accelerated model for studying osteoporosis. Muscle atrophy, well, it mirrored the effects of prolonged bed rest or certain diseases. And the cardiovascular changes like fluid shifts and altered heart function, they offered insights applicable to conditions like hypertension or congestive heart failure.
Wow.
The key insight here really is that gravity, or its absence, it's a far more fundamental biological sculptor than we might have previously appreciated. It impacts systems from the skeleton right through to the heart.
Fascinating. And just so we're all clear on the terminology used in the sources, can you quickly define physiological space, low Earth orbit, and true space?
Sure, the source uses physiological space for the altitude where you need supplemental oxygen, about maybe 2 miles up.
Okay.
Low Earth orbit, or ELO, that's where the vast majority of these missions we're talking about took place, roughly 100 to 240 miles above Earth. Got it.
And true space. That refers to regions beyond the protection of Earth's magnetic field, specifically past the Van Allen radiation belts, where the radiation environment gets significantly harsher.
Okay, that gives us the environmental context. Let's rewind a bit, back to the very beginning of this journey. Those initial attempts to send life into this challenging environment, even before the 1965-1990 timeframe we're focusing on.
Yeah, the source traces these efforts back to really early missions. Using rockets like the Araby, they carried mice, monkeys.
Wow.
There was also an early satellite attempt, Discoverer 3, though it sadly failed to reach orbit.
There's a particular aerobie experiment with mice that sounds quite revealing about weightlessness effects.
Yes, it's a great early example. They flew mice with normal labyrinth function, that's their inner ear balance system, alongside mice that were labyrinth defective. Basically, mice unable to sense gravity cues normally.
And the difference stuck.
The normal mice were significantly disoriented in weightlessness, flailing, confused.
While the defective mice seemed far less bothered. They performed relatively well. The insight was immediate. Gravity's presence is just deeply integrated into our fundamental sense of orientation and balance.
And sadly, one of those pioneering aerobie flights, it had a tragic outcome for a monkey. Delayed recovery, heat exposure, it really underscores just how risky, how experimental these first steps were.
It absolutely does. These early missions, they were just fraught with technical challenges, unknowns.
Yeah.
Lessons are often learned through really difficult experiences. And this led eventually to dedicated biological satellite programs like Biosatellite.
Biosatellite third and third and the third. I remember Biosatellite, I had a recovery failure.
Right.
Biosatellite the second, the backup, that one was successful, carried a whole array of biological specimens. But Biosatellite the third, that sounds like it was particularly challenging.
Yeah.
And quite controversial.
Biosatellite the third launched in '69. Yes, It was a major undertaking. Carried a single pigtailed monkey to study complex neurological and cardiovascular responses. Tragically, the monkey's health just deteriorated in orbit. It died shortly after landing, even though they recovered it. This mission drew significant negative public attention, controversy.
And the source includes a detail that's quite revealing about the scientific ambition back then, maybe pushing too far, that the monkey was over-invaded with implants.
Yes, things like a testicular biopsy, a bladder catheter, multiple cardiovascular implants. That's Dr. Saunders' perspective quoted in the source. He acknowledged that in their drive to collect the maximum possible data, they might have pushed the physiological limits of the subject.
A tough lesson.
Yeah. The insight from Biosatellite III, though painful, was crucial. There were limits to the invasiveness achievable with the technology and understanding of the time. Future missions needed a more cautious, sort of phased approach.
Right.
And this controversy, it ultimately contributed to the dissolution of the Biosatellite Projects Office, though it was later reestablished in a different form.
Learning from setbacks, a vital part of science. So, moving on, how did NASA transition from these early, really challenging missions to a more structured approach during that core 1965-1990 period?
Well, the source details how NASA centers develop specialized roles. Johnson Space Center, JSC, focused primarily on human spaceflight, astronaut health.
Okay.
But Ames Research Center, ARC, where this publication came from, became the lead center for non-human space biology experiments.
Were there any others?
Kennedy Space Center, KSC, handled payload integration and some plant research. Marshall Space Flight Center often provided engineering support to ARC.
And ARC's own life sciences group, it evolved significantly over the years too, right? Adapting its structure.
Right, from supporting Apollo prep to the Biosatellite program, eventually becoming the Life Sciences Flight Projects Office, or LSFPO, and then the Space Life Sciences Project Office, SLSPO.
Reflecting bigger, more complex experiments, I guess.
Exactly. And they also invested heavily in ground-based simulations, things like the Space Lab Concept Verification Tests, the Shuttle Mission Development Test, SMD-3, basically to shake down procedures and hardware before committing to costly spaceflight.
The process of actually getting an experiment flown sounds incredibly rigorous. Especially the difference between getting something on, say, a manned shuttle flight versus an unmanned Cosmos satellite.
Oh, it was a complex process. For manned missions like the Space Shuttle STS, crew safety was absolutely paramount.
Naturally.
Experiments had to meet really stringent safety requirements, which limited certain types of procedures, certain equipment.
But unmanned flights offered more flexibility.
Right. Unmanned vehicles, like the Soviet Cosmos satellites, which were actually a major platform for U.S. biology experiments, during this period, they offered more flexibility. For experiments that might be riskier or require conditions less compatible with having people around.
And getting ready involved formal reviews.
Oh, yes. Key milestones, like the preliminary design review, PDR, and the critical design review, CDR. These ensured the science, the engineering, the safety were all signed off before hardware was built and integrated, often by KSC.
So much coordination.
The insight here is that space biology isn't just about the science itself. It's equally about logistics, safety, rigorous project management.
And interpreting the data that came back from space must have been tricky. How did they use ground control experiments to really isolate the true effects of space?
They used two main types of ground controls. This was a crucial methodology.
Okay.
The vivarium control was straightforward, just specimens kept in standard lab conditions on Earth.
Simple enough.
The synchronous control, though, that was much more complex. It was started at the exact time of launch and tried to simulate as many aspects of the flight environment as possible except gravity.
Like what?
Vibration profiles mimicking launch, temperature cycles, light-dark cycles, atmospheric composition, even how long they were confined. By comparing the flight data to both controls, they could better isolate the effects uniquely attributable to microgravity and radiation, separate from the stresses of launch, landing, or just being cooped up. That insight, the need for rigorous ground simulation, was essential.
That makes perfect sense. You have to peel away all the other variables. Okay, let's get to the findings. What did they actually discover about how microgravity affected life? Starting with, say, bone and muscle.
This was a major focus, especially with rodent studies on those Cosmos missions. They consistently found that rats exposed to microgravity showed significant decreases in bone stiffness and formation. Muscle mass also decreased. And this atrophy was aggravated by the hypokinesis, the reduced physical activity that just naturally happens in weightlessness.
But you mentioned something earlier about centrifuges fixing some of those issues.
Yes, this was a really key experimental design used on missions like Cosmos 1129 and 2044. Some animals were housed in cages that were periodically rotated on a centrifuge, providing artificial gravity.
And the result?
The insight was profound. The bone and muscle issues were prevented, or at least significantly reduced, in the centrifuged animals.
Really?
Yeah, it provided compelling evidence that the effects were directly caused by the absence of gravity, really highlighting gravity's fundamental role in maintaining musculoskeletal health. They also studied related things like calcium metabolism and how the bone matrix matured.
What about the cardiovascular system, those fluid shifts you mentioned?
Studies, notably on rhesus monkeys and rats flown on missions like Cosmos 1514, they explored these responses. They looked at changes in heart function and that well-documented shift of fluids from the lower body up towards the head in microgravity.
And going back to the disorientation in those early mice, what did later neurovestibular studies find out?
They built on that initial observation. The orbiting frog otolith experiment, OFOA for instance, it showed altered neural control processes in the inner ear's gravity sensors, the otoliths.
In frogs. Interesting.
Yeah. And studies on rhesus monkeys looked at complex reflexes, like the vestibulo-ocular reflex, how eyes track with head movements on missions like Cosmos 1887 and 2044, investigating how the brain adapts, or maybe fails to adapt, to the confusing lack of gravity cues for balance and vision stabilization.
Plants also rely heavily on gravity to orient themselves, right? Did they grow differently up there?
Oh, absolutely. This is one of the most visually striking findings. Experiments like the STS-34GHCD experiment, that's gravitational hardiness and cell differentiation with mung bean, oat, and pine.
Yeah.
Roots that would normally grow downwards towards gravity. They grew upward toward the light source instead.
Whoa.
This supported the hypothesis that lignin synthesis, what gives plants rigidity, is reduced in microgravity. Another study on biosatellite dissecant with wheat seedlings noted that their coleoptile height actually increased after the plants were returned to Earth gravity, suggesting complex changes in their growth regulation in space.
So gravity is like a developmental signal for them?
The insight, exactly, is that gravity isn't just a physical force for plants. It's a fundamental developmental signal.
What about life at the smallest scales? Or development from scratch?
Well, early experiments included things like sea urchin egg development on Gemini the Third, amoeba cell division on Biosatellite Second, showing that some basic processes could occur.
Okay.
Later, on STS-29, the SFP study used rats to look at bone fracture healing and overall development in microgravity, investigating how complex tissue processes proceed without those normal mechanical loads.
Circadian rhythms, our internal clocks, they're crucial. How are they affected?
This was studied across various organisms: pocket mice on Skylab 3, fruit flies on Skylab and Cosmos, the biosatellite the Third monkey, even fungus like Neurospora crassa on STS-32.
What did they find?
The findings suggested that fundamental circadian processes can persist in space. But the monkey study provided a really significant insight. While some rhythms continued, there was evidence of internal desynchronisis, where different biological systems within the same organism got out of sync, which is, a major concern for astronaut health on long missions.
Immune function is another big factor for astronauts, I imagine.
Definitely. Studies on rats, like on Cosmos 1887, began exploring this area, investigating changes in immune cell responses, surface markers, laying groundwork for later, more in-depth studies.
And aging. Did they see any accelerated aging in space? Was that a concern?
Fruit fly experiments on Cosmos 782 and 936 initially didn't show appreciable effects on development or aging in the absence of gravity.
Really? That's
It
surprising.
was at the time, yeah. And it prompted further studies to understand why, or perhaps if, that was truly the case across different species and longer mission durations.
So microgravity fundamentally alters biological processes pretty much across the board. Now let's switch gears to the other major challenge, radiation. How did studying space radiation effects differ from studying radiation on the ground?
Well, space radiation is like a cocktail of different particles and energies you just don't find on Earth. You've got charged particles, neutrons, ionizing photons.
Right.
Ground-based studies often use nice, uniform sources. In space, they needed accurate dosimetric measurements, not just the total dose, but things like the fluence, how many particles pass through, their charge, velocity, and something called linear energy transfer, or LET.
Right.
It's a measure of how much energy a particle deposits as it travels through tissue. The insight was that understanding space radiation required entirely new ways of measuring it in situ, right there in space, and correlating that measurement to biological effects.
And what biological effects did they find that were specifically linked to this unique space radiation?
Some concerning findings emerged, yeah. Rats on Cosmos 1129 and 1514 showed detrimental effects on their retinal tissue. Pocket mice on the Apollo 17 BioCore experiment exhibited necrotic lesions, dead spots, on their scalp and skin, likely from hits by high-energy heavy particles. Biosatellite 2 experiments found evidence of genetic damage in fruit flies and wasps and mutagenic effects in fungus. Tumor immunotherapy was even explored as a potential research area, sort of leveraging the unique radiation environment.
And they even flew dedicated hardware just to measure the radiation itself, didn't they?
Yes, precisely. Cosmos 1887, for example, carried sophisticated radiation dosimetry experiments. They used different types of hardware to capture the full picture.
Like what?
Things like plastic nuclear track detectors, PNTD stacks, and nuclear emulsion to track individual high-Lebbe particles, thermoluminescent detectors, TLDs, to measure total absorbed dose, and activation foils to measure neutron exposure.
So multiple tools for a complex job.
Exactly. The insight here was that a single sensor just wasn't enough. You needed multiple types of detectors to characterize that complex space radiation field properly.
It's clear they needed highly specialized equipment, not just for measurements, but to house the organisms, run the experiments. Can you paint a picture of some of this specific hardware?
Absolutely. A key piece developed during this era was the Research Animal Holding Facility, or RAHF. RAHF, used on Space Lab missions like STS-51B and SLS-2.
Okay.
This was designed to house rodents and small primates for extended periods. It had modular cages, automatic feeding and watering systems, activity monitors.
Yeah.
Pretty sophisticated for its time.
And I understand even this state-of-the-art hardware had some unexpected quirks.
Oh yeah, spaceflight always throws curveballs.
Yeah.
The source mentions issues with the RAHF like water leak alarms being triggered by enthusiastic monkey drinking patterns. Really?
Yeah.
Yeah. And waste elimination orientation problems causing, well, fouling and microgravity. These seemingly small details highlight the real-world complexities of designing for zero-G.
True. What about for plants?
For plants, the Plant Growth Unit, or PGU, was developed, used on shuttle mid-deck missions like STS-3. This was a self-contained unit with controllable lamps, timers, sensors, fans, all to support plant growth experiments with species like wheat, pepper, and mung bean.
And for the unmanned Cosmos missions, the hardware often needed to be completely autonomous for months, right? Stand alone.
Exactly. Hardware, like the small self-contained temperature recording system mentioned in the source, was crucial, capable of storing data for the entire mission duration until they could get it back.
Ingenious.
And back on Biosatellite III, with the monkey, they had pioneering systems like the primate psychomotor test system, testing memory and coordination, using food rewards, and even an automated urine transport system and analyzer to collect and process samples right there in orbit.
Wow.
The insight really is that every scientific question required novel, often finicky engineering solutions, and learning from hardware failures was just as important as the scientific findings themselves.
This wasn't just U.S. science either, was it? A truly remarkable aspect highlighted in the source is the significant international cooperation, particularly with the Soviet Union during a period of pretty intense geopolitical rivalry.
This was a powerful and, frankly, very productive collaboration. especially through that Cosmos biosatellite program.
What made that cooperation so beneficial? Even necessary, perhaps?
Well, the source points to the reality that both countries had limited access to spaceflight opportunities. It was expensive, difficult.
Sure.
Pooling resources and flight slots allowed for larger, more complex missions than either could easily achieve alone. They also benefited from standardizing procedures, exchanging data, acknowledging that different methodologies, like, say, how they measured blood pressure, needed to be reconciled for the comparisons to be valid.
The scientific challenge transcended politics.
The insight was exactly that. The scientific challenge of space biology was just bigger than national borders.
How did the U.S. involvement in the Soviet Cosmos program evolve over this period? Did it start small?
Initially, yes. U.S. experiments were often self-contained carry-on packages. They didn't need much integration with the Soviet spacecraft system.
Yeah.
But over time, as trust and capabilities grew, especially starting with Cosmos 1514, which was the first primate mission involving U.S. hardware integration, they began integrating U.S. instruments directly with Soviet spacecraft data systems. Joint hardware development became increasingly common.
There's a quote from Dr. Winter about the U.S.-Soviet collaboration that really captures the spirit of it, isn't there?
Yes, he described it as this fascinating blend. On one hand, competition for success and public recognition. But alongside that, a deep shared goal of avoiding disaster by pooling resources and information.
Neither side wanted a failure.
Exactly. That mutual vulnerability fostered this unique form of collaboration.
And Rodney Ballard's anecdote about Dr. Iliam, the Soviet program lead, recounting overcoming obstacles like oxygen increases, rising temperatures, wire breakages, it really brings the human side of that collaboration to life.
He does. His pride in the joint efforts, leading to his dream of an international biomedical laboratory in space.
Yeah.
It just shows how facing those incredibly difficult technical and biological challenges together forged this powerful bond, a shared vision for the future. The insight is that science really can be a powerful bridge, even in the coldest political climates.
Beyond the Soviet Union, the source also mentions collaborations with France, the European Space Agency, ESA, Canada, Japan, even the U.S. Department of Defense. It truly became a global effort over time.
Indeed. Space biology, almost by its nature, requires international cooperation to maximize those limited opportunities.
So, pulling all of this together, this immense effort spanning decades, studying everything from fungi to primates, what's the overriding conclusion from this era of space life sciences at Ames? What's the big takeaway?
I think the key takeaway is the sheer breadth of biological systems and organisms they investigated, and the fundamental nature of the questions they tackled. They established, really unequivocally, that space isn't just a vacuum. It's a profoundly active environment for living systems.
Impacting everything.
Pretty much. From cellular processes and genetics right up to whole organism physiology and behavior.
And they gathered invaluable data, often despite significant challenges, hardware failures, difficult recoveries, the whole political backdrop.
Absolutely. And the source highlights that later studies, particularly on space lab missions like SLS-2, advanced the field significantly. They allowed for more complex in-flight procedures like blood draws, even dissections.
Which helped separate out effects.
Exactly. That was crucial for gaining the insight needed to differentiate effects happening during spaceflight from those that might be part of the stressful landing process or just readaptation back to gravity.
This historical work wasn't really an endpoint, was it? It was truly the foundation for everything that followed.
Precisely. This whole era laid the essential groundwork for planning longer duration missions, thinking about lunar bases, journeys to Mars, by providing the first real insights into the biological hurdles. And it hinted at the necessity of continued, maybe even deeper international efforts in the future. The insight is that building towards future human space exploration really starts with understanding life's limits and its resilience.
It's an incredible legacy of discovery, built, as you say, brick by biological brick, often through sheer perseverance. We've journeyed through decades of groundbreaking space life sciences.
From those very first risky rocket flights with mice,
to complex, internationally collaborative missions studying everything from fungal rhythms and plant growth to primate physiology.
And the knowledge gained. It continues to inform space exploration and, importantly, Earth-based biology today.
Indeed. And as we wrap up this deep dive, maybe consider this. The very challenges posed by the harsh environment of space, that lack of gravity, the presence of complex radiation, they aren't just obstacles to overcome.
Hmm.
By studying how life responds under these extreme conditions, we gain insights not just into what it takes to live off-world, but into fundamental biological processes that could unlock entirely new ways to understand and maybe even treat diseases right here on Earth.
Yeah.
So what other extreme environments, perhaps closer than you think, might hold similar keys to understanding life's incredible resilience and adaptability?
That's something truly worth pondering.